Hi Hires, we are looking at KDS7 speciation in DNA in the genome. So a lot of this builds very carefully and clearly on the National 5 level, and it would be worth you just reviewing that to make sure you're okay with it, or if you go through this and find this incredibly difficult, go back, look at the Nat 5 and then come back to it, because there are some very much more detailed terms, but overall it's quite a clear progression. Okay, so this is a new one really, the idea of a gene pool. So a gene pool is all of the genes that are present. Now that means not just what genes are coding for, but how many alleles there are for each of them, how many of them are present. It would describe it in terms of frequencies. So in this particular gene pool picture that we've got over here, we can see that we have three particular alleles. We've got a green allele, a blue one and a red one. But we would also say that we have got much higher frequency of the green than we do of the blue, and the red is a very low frequency. Okay, so every allele that we have, that is the gene pool. So if there is absolutely no selective pressure, so there's nothing that's pushing any one type of these alleles to have a particular advantage, then what you would expect is all of the alleles just get tipped in the gene pool and then pull back out again at the same frequency as they were every time. Okay, that would be if there was no pressure at all. If you're interested in this, there is something called genetic drift, which does impact even if there's no pressure, and it's an interesting one. Okay, so as I say, this builds very much on the Nat5. So the speciation that you had at Nat5 is not bad at all. This is our definition. It's a group of individuals that can interbreed to produce fertile offspring. It's not bad. It is a specific technical thing that we're using to define species, and it's not perfect. So at higher, we've added in another bit, which is to say that they do not breed with other groups, or don't tend to breed with other groups. Speciation is still the same. It's the development of a new species. And the overall how you do it, pretty much the same. There's just some more technical terms. So if we start off with our population, this is my random little dudes that I've drawn. These are mugwumps or whatever you want to call them. So inside this, their gene pool would be whatever it is inside them at the moment. So obviously in terms of colour, they're pretty much all blue, and they all seem to be looking at the same thing, but I'm not sure why. Well, I do, it's because I copied and pasted. Okay. So then you need to isolate, and that is exactly the same as you had at Nat5. You said that you had to separate them out. So this is us isolating with a barrier. And our three types of barrier are geographical, ecological, and behavioural. Okay, so we have here for this population a geographical barrier. Okay, we're putting up mountains that look suspiciously like a greyish Toblerone. Okay, so what we've done here is isolate two gene pools, and now the isolation has to be complete. What you're looking for is no movement between the two populations. If there is movement between the populations, so if you have like this dude just sneaking around the side of the mountain, and that would mean that you've now got gene flow. And what we're trying to do is prevent gene flow. That's what has to happen. We have to have no movement of genetics from one set of genes to the other set. Okay, so now we need variation, because each subpopulation at the moment still has exactly the same gene pool. We've just put a big mountain in the middle. So what we now need to do is variation needs to start kicking in. And that's going to be any new variation has to come through mutation. So this could be taking quite a long time. And what we're getting is a drastic set of variation inside the gene pool. Drastic, I'm saying drastic because, you know, this little dude has just lost both his eyes, and this one has a random stompy foot. And I don't know what's happened to this guy's eyes, and this one has become quite tall. So we've just assumed that we've now got different variation in each of them, and that is important. Okay, and then we're assuming sexual reproduction within this group of whatever they are, and that means that we're mixing the alleles constantly. We now have separate gene pools. Finally, we then need to have selection. Now the selection has to mean that you have different pressures on either side. So it has to be something which is not going to push for the same alleles to be selected. So what I'm saying on this side is, let's say that our tall individuals are favoured because they can see over the tall grass. Okay, and on this side, which is colder, it is the little short, hairy, fuzzy ones that are better prepared. Okay, so they will increase. The rest of them We'll just assume they all died out. And eventually that would cause speciation. But it has to be an extremely large number of generations to get to speciation. You can see the evolution before you get to speciation. If you bring them back together and they can't produce fertile offspring, then by our simple definition of it, we would say that these are different species. And that's about it. So basics are still the same, and this is all right. Okay, we have isolation, we have mutation and variation, and then we get selection to cause the speciation event. But there are two specific forms of speciation that you need to be able to define for hire, and this is allopatric speciation and sympatric speciation. They're just big words that you need to know. Allopatric speciation means specifically that we are looking at a physical barrier. So that's a geographical barrier is going to cause allopatric speciation. So this is a physical barrier stopping gene flow. So it is like your mountains and the seas and deserts and physical things. Okay, that's it. That's all we need to know, geographical. Sympatric is the tricky one, because sympatric speciation is where the populations manage to stay in the same geographical location and still evolve. So what's happening here is we have barriers that are preventing them from breeding even though they live in the same place. So there is a clever one which we'll just mention here, which is plants. So plants can actually survive mutation events called non-disjunction, where the spindle fibers don't function properly. So if you have a cell, if I show you a cell here, okay, and we normally have our cells all joined up at division. Okay, we've got our spindle fibers kind of pull out from here, and normally what we'd expect is one would go this way and one going this way. That's normal. But if this spindle fiber breaks here, then what happens is we get two going this way, and so we double up on a chromosome. Now that is not super common. As humans, we can't deal with that at all well, but plants can manage it even if they break all of the spindle fibers on one side, so they get a double dose of all of their chromosomes. And that can mean that you end up with something that can't breed with everything round about it in just one mutation event. But they're still in the same place. If they then go through vegetative propagation, which plants can do, so they just produce clones of themselves, they've actually produced a set of plants that can breed with each other, but not with everything round about it, in one step. Okay, so it's possible to do it in some ways very quickly. So to show an example of what we're looking at here, so if we remember from selection, if you have the kind of range of a factor, and this would be the mean at this point here, if you select for top and bottom ends of this, the extremes on either side, then what happens over time is we get this kind of happening to the whatever factor you're looking at, and it actually starts to split into two different means. So this is disruptive selection. So let's imagine that we have some flies that are feeding on apples. Okay, let's make the factor along here the color of apples. Okay, so we've got red at this end and green at this end. Okay, so this is red and this is green. If you end up with a group of flies, these ones here, which only feed on the green part, okay, and this group of flies which only feed on the red part, then they stop breeding with each other. And so what you end up with is a group of flies eventually which cannot crossbreed with the other group of flies. So you can have a form of selection, disruptive selection, which is for the preference for colour, that could eventually end up with giving you two totally separate species and in the same place, because they're maybe on the same tree, so sympatric. This is just another picture of it. The idea being that we have plants here, and in one of them we've got a geographical barrier right down the middle of it, blocking them, allopatric, and here we have a reproductive isolation going on, so we have two different subpopulations which are not managing to breed with each other. And that's your lot.